right, so we've talked about the breakdown of organisms into domains and kingdoms, and that we come up with these levels. So today we're going to talk about how do we devise these classification systems? How do we determine what organisms belong to what category? So the old way to do it, before we really knew a lot about evolution, before we knew as much about DNA technology, things like that, we just based it on morphology. Early systems just based it on what they look like. So for example, in this picture, we can see that the limpet and barnacle are classified as being related to each other, more closely related than they are to the crab, because they both have shells. But now we actually do it using evolutionary classification, or what's called phylogeny. So what we're looking at is evolutionary relationships, like what are the ancestors? Do they share a common ancestry? You know, where did this organism come from? And so we base it on the same things we use when we study evolution. I'm not going to spend a lot of time because we've already talked about these: comparative anatomy, embryology, DNA, and fossils. And what we make is called a cladogram, which is like an evolutionary tree that shows the relationships between the organisms. And so it uses something called derived characters or derived characteristics that show when or at what point in evolution these different characteristics appeared. So for example, looking at our original classification of the crab, barnacle, and limpet on the left and our current classification on the right, you can see that in our cladogram, we're basing their subdivisions on an evolutionary relationship. So they all have tiny free-swimming larvae, but then the crab and the barnacle both have segmentation and skeletons that molt. And we also believe that the crab and the barnacle share a more recent common ancestor than either of them share with the limpet. So these breaks or nodes are where they share a common ancestor, and these are what we're talking about in this cladogram, these derived characters, these characteristics. So nobody prior to this point has this, but everybody past this point does. And so we use, again, running through these very quickly, homologous structures. Remember, this is like the bones in our arm and the bones in the wing of a bat, how they have similar underlying bone structures, similar embryological development, similar DNA, RNA, amino acid sequences. So we actually look at the DNA of organisms to determine their relationships. This is homologous structures, you may remember from our last chapter. This is embryology, looking at the embryological development of different organisms. And so how do you read one of these? So here's a cladogram. These are your derived characteristics. That's what the derived characters are. And these, called nodes, these are where each of these organisms sort of branch off. So we would call the lancelet, he's actually called our outgroup, because he's considered the least related to all the others. He sort of branches first before any of these other characteristics evolved. So what characteristics do all salamanders have? So to answer this question, what you would do is, I'm just going to change my color here, is you would track the salamander back to the beginning. This would be, tracing back in time to the original ancestor. Notice this one doesn't actually show time, so we don't know when these traits appeared. Some of these will actually have time periods, but this one doesn't. Anyway, so the traits that the salamander would have would be four-legged locomotion, jaw bones, and a vertebral column. Everything that he hits when I track his line back to his ancestry. What characteristics do only wolves have? Well, nobody but wolves has hair. So that would be a characteristic that's unique to wolves. Also, I talked to you before about knowing how to figure out relationships. Like if I said, hey, is the salamander more closely related to the wolf or to the grouper? We did this in our last chapter too. A lot of people would mistakenly say, oh, he's more closely related to the grouper because look, he's right next to him. But that's not how you read this. Remember, you're tracking back who shares a more recent common ancestor, which is where their lines merge in the cladogram. So the wolf and the salamander here, but the grouper and the salamander, change my color again here, are all the way back here, right? That's where their two lines merge together. So they have an ancestor further back. In fact, the salamander and the wolf have more characteristics in common. The grouper and the salamander only have jaw bones and vertebral column, but the wolf and the salamander have also four-legged locomotion. So that's how you read one of these. This is another one just showing primates. I don't like this one because it kind of puts these here, and they normally should go here in between the organisms. Okay, so how do you make a cladogram yourself? So first of all, this would normally be filled out for you unless it was something that you would already know. So we've kind of talked about some of these things, but we haven't talked about all of these, so I wouldn't expect you to know how to fill this out. So we're going to put a check mark, or we can put a plus or whatever, next to characteristics that an organism has. So for a eukaryote, the amoeba, mushroom, fly, and dog all are eukaryotes. Bacteria is actually a prokaryote. Next, multi-celled. Well, the mushroom, the fly, and the dog are all multi-celled. Appendages, which are legs, flies and dogs have, and then a backbone, only the dogs have. All right, now how do we turn this into an actual cladogram? So what you want to do to start, step one, is you want to look for either an organism that has no characteristics, or sometimes you'll have a characteristic that every single organism has. So in this particular cladogram, I see an organism with no characteristics, the bacteria. He has none of the traits, right? So he's going to be our outgroup. He's going to go first. Now, if we had, on the other hand, a trait that everybody shared, it would go back here. Like, for example, let's say I added a trait here. Let's say I added living as a characteristic, right? Living. Then everybody would have that trait, right? 1, 2, 3, 4. Then living would go back here. Living. It would be my first thing on this cladogram because everybody in the cladogram has that. But in this cladogram, we're actually starting with the an organism. Okay, so now the easiest way to do these, and it's easier digitally because you can still erase if you make a mistake, is we're going to cross off the bacteria because we've already included them. So now that we have an organism, now we want to look for a characteristic. So what characteristic came right after bacteria? Now, what we're looking for basically is a characteristic that everybody has. So whenever we're looking at characteristics, we're looking for all check marks. And wherever we're looking at organisms, we're going to be looking for all zeros, negatives. Let me show you what I mean. So we've got our bacteria, we've crossed him off. What's now the characteristic that everybody else has? Now that bacteria aren't in our cladogram, or they're already placed in our cladogram, we can see that the characteristic of eukaryote is a characteristic that everybody has, right? Check, 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 because bacteria we already included. So since everybody past bacteria has that characteristic, we can put that here. Usually we spell it with a K. I have it with a C here, but it can be spelled that way. It's more commonly with a K. All right, so now that we've done that characteristic, notice I've crossed that off. Now we basically go back and forth. So we've got an organism, now we've got a characteristic. So now we should be going back and looking for an organism again. Sometimes these can get more complicated, but we're going to keep them simple. So what are we looking for? We're not looking for an organism that has everything. We're looking for an organism that has no other characteristics past eukaryote. So that organism would be the amoeba. Notice how he does not have any of the remaining characteristics, only that he's a eukaryote. So if we put amoeba here, notice how our cladogram is starting to take shape. Our amoeba has just the one trait of being a eukaryote. Our bacteria had none of the traits. So what are we going to do next? Well, we're going to cross off our amoeba just so we can keep track. And now we're going back. We just did an organism, so we want to look for a characteristic. Is there a characteristic that everybody that's not in the cladogram yet shares? And that would be multi-celled, right? Everybody that's left has that characteristic. So multi-celled is going to be our next characteristic. And we're going to cross that off. So multi-celled, check. Now going to go back to organisms. Is there an organism that has eukaryote multi-celled, but they don't have anything past that point? And the answer is yes, the mushroom. He's next. He does not have any further characteristics. So the mushroom's going to go here. We would cross him off. Now we need our next characteristic. So our next characteristic that everybody has is going to be appendages. Now our organism, that's going to be the fly. And then our last characteristic is going to be a backbone. And our last organism is going to be the dog. Oh, and technically, you can just put him at the end here. Dog. You don't have to make a separate line. Now, if you did this correctly, you should double check yourself. pick an organism in your cladogram. So let's say I pick, the fly. If I did this correctly, the fly should have appendages, multi-celled, and eukaryote, but not a backbone. Does that match the fly? Yes. Appendages, multicellular, and eukaryote, but does not have a backbone. So you can double check yourself. The organism at the end should be the one that has the most check marks. The organism at the beginning should be the one with the least number of check marks. That's our out group. And the more derived characters, they have in common, we would consider them more evolutionarily related to each other. So that's how you make a cladogram. This is a picture showing a kind of a breakdown of the different domains, and you can see how they sort of split into all of our different kingdoms, the protist plants. You can see fungi here, this is animals here, and so this is sort of a phylogenetic tree, they call that. It doesn't actually show the characteristics. Okay, and that brings us to our very last thing really quick, which is reading a dichotomous key. Here's the thing with a dichotomous key. You're never going to go wrong if you follow the rules, and the most important rule is to start from the top. Don't just look through all the things in the key and find a characteristic that fits, because that can mess you up. So it's easier to show you one than to explain it, but you can buy a book of like a dichotomous key of, say, bird classification or mushroom classification or whatever. It'll have characteristics in pairs, and it's sort of an either-or thing. The organism's only going to have one of the two, and based on the one it has, you go where it says. So we start with 1A and 1B. Always. Do not just randomly look through here and go, oh, jellyfish. This is a jellyfish, so I'm done. Because, yeah, you know that's a jellyfish. But what if instead of jellyfish, they had names here, scientific names? You wouldn't know its scientific name. That's the whole idea of the key, is you're trying to figure out its name. So you're going to always start at 1. So tentacles present, you're going to go to 2. Tentacles absent, you go to 5. So there's only 2 options. One of them's going to be true. One's going to be false. So if I look at this one, tentacles are present. So I'm going to go to 2. I go to 2. 8 tentacles, it's an octopus. More than 8, I go to 3. So I look, it actually does have 8. So this one is an octopus. Let's just try one more, and then I'll show you one with leaves that's a little harder. Okay, so again, we're going to start from 1. Tentacles present, go to 2. We're doing this one. So it does have tentacles, right? These are tentacles, so we're going to go to 2. When I go to 2, it has more than 8. If I look at all of these, there's more than 8. So now I'm going to go to 3. Do they hang down or are they upright? Oh, they're upright. So this must be a sea anemone. So you see how this works. Again, you can kind of cheat on this one because you might already know what a sea anemone looks like. But look at this one. So now you've got a bunch of leaves. So let's try classifying this leaf. You don't know what that is. I mean, unless you're a botanist, you probably don't know. So start at 1. Needle-like leaves are non-needle-like leaves. So needle-like leaves would be like these. So it does not have needle-like leaves. So we are going to go to 3. So now I go down to 3. So I'm skipping this. Go down to 3. Single leaf are compound leaves. That is, in picture A, definitely a single leaf. So I'm going to go to 4. If I go to 4, smooth edge or jagged edge, it definitely has a smooth edge, so I'm going to go to 5. Leaf edge is smooth or leaf edge is lobed. So lobed would mean like this. That's what they mean, because it's not jagged, but it's got loops. It's definitely smooth, so this must be a magnolia leaf. Now notice, you can't just look through and just pick, like, oh, starting anywhere in the middle and then just work your way down. You always need to start from the top and work your way down. Because sometimes you might have, like, smooth and lobed in more than one place. But if you were never sent to step 5, if it said to go to 6, then it's definitely not going to be either one of these. So that's how you're going to use a dichotomous key. And I actually have a practice one for you to try with some sharks, because you're not going to know the names, their scientific names offhand. So it'll be a good practice with using this technique to read a key.